So we're going to talk about root hairs for a moment. The function of the root hair is to collect water and mineral nutrients present in the soil and take this solution. I want to pause on that word for a moment. This solution. That means that the mineral nutrients are dissolved in the water. That means that they are solutes in the water. So this solution up through the roots to the rest of the plant. So mineral nutrients are traveling dissolved in water. That phrasing is usually worth points. Mineral nutrients are traveling dissolved in water up the plant through the xylem. So root hair cells are adapted for water and mineral uptake. So root hair cells are adapted for this by having a high surface area to volume ratio, a thin cell wall which decreases the distance over which diffusion must occur, many membrane transport proteins to be able to handle the uptake of mineral ions, many mitochondria for the production of ATP to allow for these membrane transport proteins in the event that they are active transport proteins. No chloroplasts because this is underground. This is a root hair cell. And last I checked, there's not that much sunlight underground. And no cuticle. A cuticle is a waxy barrier. It prevents the loss of water or the movement of water through that cuticle layer. So if our root hair cell had a cuticle, that would be a watertight barrier in a root hair cell that is designed to allow water to enter. That would be very counterproductive. So these are how a root hair cell is adapted to its function. Its surface area to volume ratio is high. Surface area is directly correlated with the rate of diffusion. So higher surface area allows for greater rate of diffusion. Thin cell wall, decreased distance over which to diffuse those substances, so increasing rate of diffusion. Increased membrane transport proteins. That means that you have effectively a greater surface area for facilitated diffusion and active transport. You can transport more of this stuff. So I've already said those things, but I wanted to reiterate because it is important, and understanding why can get you additional points. Explaining why and being able to explain why is important. So the mineral uptake. Anything that interferes with the metabolism of these root hair cells can reduce the mineral concentration in plant cells. And here's the thing. The mineral concentration in roots can be up to 10,000 times greater than the concentration of minerals in the surrounding soil, thanks to this active transport. So what's the water going to do if the mineral ion concentration in the root hair cell, if that salt is so much higher in the root than in the surrounding soil? What does water do? Right here, there's going to be this big flashing bubble, in case I remember to edit, saying, okay. Now, the movement of water through plants is ubiquitous. These are the same three pathways throughout the entire plant. But we're going to discuss them right now in reference to the root hair cell. These also apply to the entire plant. They apply to the stem. They apply to the leaves. They apply to every other part of the plant that you care about. So stems and leaves, yeah. All right? But for now, let's discuss them in terms of the root. We have our apoplastic pathway. The apoplastic pathway is the pathway that does not pass through the cell. It does not pass through the cytoplasm. It is a non-cytoplasmic pathway. So it passes only through the cell wall. Now, the xylem vessel does not have cytoplasm, which means that the xylem vessel is actually an apoplastic pathway, interestingly enough. So the apoplast pathway, water and dissolved ions move through the cell walls between the cellulose molecules. So to cross into the xylem, it goes through the apoplast pathway. It must cross into the xylem in that fashion. And at the beginning of the xylem vessels, there is no lignin. And at the ends of the xylem pathway, in the leaves, there is no lignin, because that assists in making that xylem vessel watertight. Now, the symplast pathway. The symplast pathway is moving with the cytoplasm. Water moves into the cell through the plasma membrane and then moves through the cytoplasm from cell to cell through the plasmodesmata. The vacuolar pathway is where water goes through the cell's plasma membranes into the cytoplasm and then into the vacuole, and then out of the vacuole, and then in through the plasmodesmata, and then into the cytoplasm, and then into the vacuole, then out of the vacuole. Continuing on like this. So the symplast pathway is moving through the cell, moving through the cytoplasm. The vacuolar pathway is a subtype, if you will, perhaps, of the symplast pathway, where you're also going through the vacuole. Now, the movement of these pathways will all move from the epidermis, the epidermal hair, all the way through, until you reach the endodermis. At the endodermis, the apoplastic pathway ends. And we'll talk about why in a moment. The endoplasmic pathway ends, but with the symplastic or vacuolar pathway, which here it's labeled transcellular, but vacuolar is the Cambridge preferred method of naming it, they can continue as normal through the endodermis into the pericycle and then into the xylem. The endodermis must be shunted from the apoplastic pathway into a cell, and then through the symplastic pathway into the vascular bundle. The reason for that is the Casparian strip. The Casparian strip is a band of suberin named after R. Caspary, some dude, some science dude. So it's a band of suberin. Suberin is a substance that's not permeable to water. It creates this it prevents the movement of water through the cell wall. And when that water hits that suberated band, it must be shunted into the symplastic pathway. So that suberated band allows for everything to be shunted through the endodermis into the pericycle by the symplastic pathway. This is going to be important when we talk about maintaining a solute potential gradient by means of the movement of sucrose out of our phloem, through our pericycle, through our endodermis, through our cortex, into our epidermis. So, because of this movement of sucrose, we need that water to be shunted into the endodermis, because otherwise water doesn't have to follow salt. By forcing it through the endodermis, we force water to follow salt from soil to vasculature. We are creating a choke point here. And we are forcing this sucrose gradient to be part of the water potential gradient chain that moves water from the outside of the plant to the inside of the plant. Now, understand that sucrose concentration is highest in the phloem because it just came from the source of that sucrose. And active transport is pushing phloem sucrose, out at lower and lower and lower concentrations as it moves outward to the epidermis. So water is going to follow this salt to the higher concentration of sucrose wherever it can. Once water is forced through this suberated band, it can return to the apoplastic pathway or stay in the symplastic pathway if it so chooses. By chooses, I mean whatever is the least resistance path. There is no problem there. as long as it goes through the endodermis in the first place. Because we can't control sucrose concentrations outside the cell. We can only control sucrose concentrations inside the cell. Now, let's talk for a moment about root pressure. Root pressure is the pressure that's created by all this water flowing into the root. All that water that comes in takes up space. Water is not some ethereal substance that takes up zero space. If you drink a lot of water really fast, you get full. So water takes up space. You can perform that experiment at home, but please don't drink too quickly. That can be actually a little bit dangerous. So root pressure is the pressure that's created by the water potential gradient within the cells of the root system that causes the sap to rise through the plant stem to the leaves. Root pressure occurs in the xylem of some vascular plants when the soil moisture level is either high at night or when transpiration is low during the day. So that's root pressure. We can see root pressure if we were to cut a tomato plant off at the root, at that transition zone. We would see not like a Kill Bill style volume of blood shoot up out of the stump, but we would instead see sort of a slow ooze of sap come out of the base of the plant. That is root pressure. Now, yes, that pressure does make a difference. It is somewhat significant, but it is not all the pressure that you need to get water all the way up the plant. It might get you to maybe ankle or shin height in terms of plant height. But ankle or shin height is not as high as plants go, in case you've ever been, I don't know, outside. But this is the internet, who knows? Alright, so what we need from that point on is actually going to be something that is brought about by the continuous xylem, the cohesion tension theory. Transpiration pull. So, cohesion, if you remember Unit 1, way back, like 3 or 4 videos ago by now. I'm not sure of the editing at this point. Cohesion is going to be due to hydrogen bonding between the polar ends of the water molecules. When transpiration removes water from the leaves, that resulting low water potential in the leaves will pull nearby water molecules into the leaves. And because of that attractive cohesive force between molecules, this transpiration pull translates into cohesion tension. And on the adjacent water molecules, and those adjacent water molecules will be pulled to fill in that space. In the adjacent water molecule, fill in the space that gets left behind. That continues all the way down a continuous column of water. As long as there are no air bubbles or no side steps in that xylem to break the chain. So this also is why xylem must have lignin in order to strengthen it. Because if you have ever had bubble tea, for example, or perhaps too thick of a smoothie, you know that if your straw is not wide enough for the bubble, or the ice cube, or the ice cluster at the bottom of the tea, your straw is going to collapse when you start providing suction. So your straw collapsing is because your straw is insufficiently has insufficient structural integrity. The lignin provides additional structural integrity and allows these. Pause the video real quick.